Our first change maker today evening is Vighi, a Ph.D. student in Department of Bioengineering working under the supervision of Professor Codruta Ignea. With Asia, we are going to discover how something small as a yeast cell can help enhance the production of some of the key pharmaceutical drugs.
I love plants. From the daisies on the side of a highway to the massive trees on the side of a hiking trail, plants are incredibly diverse and resistant organisms. But by far, my favorite thing to do is to watch the plants grow in the greenhouse my dad built in our backyard years ago. And although it may seem slow and uneventful on the outside, there's a lot going on inside of the plants for them to mature from seed to seedling to maturity. Plants will perform photosynthesis to convert solar energy and carbon dioxide into the sugars and the energy that they need to grow. And this allows them to develop and mature as organisms. But of course, for any organism to thrive in their environment, they need more than just energy to grow. And so, although these key processes are part of their central metabolism, plants will also have a specialized metabolism, producing thousands of other low molecular weight compounds. These compounds are typically produced in very low quantities, but they perform very important roles for plants in signaling, in protection, in pigmentation. And for us as people, they have a lot of useful applications as well, in cosmetics, biofuels, pharmaceuticals, as flavors, fragrances, and a lot more. And so today we'll be taking a little bit of a closer look at one of the compounds from this pharmaceutical category, Tanshinone IIA, or T2A, which is naturally found in the roots of a red sage plant as a specialized metabolite produced in very low quantities in that natural host. However, shows a lot of promise in the treatment and prevention of many different cardiovascular diseases. And if we were to try and cultivate enough red sage in order to treat the millions currently living with heart disease, we would need a red sage monoculture covering an area of about 5.5 times the size of Montreal. This is extremely resource intensive and simply unsustainable. Although as a promising compound, of course, we do want to find a way to produce and obtain it. An alternative method that's often posed is chemical synthesis, which allows you to control the pathway. However, as a lot of secondary or specialized metabolites are very complex, controlling exactly which oxygen attachment you're getting at each different position on the molecule can be quite complex. It can be difficult. The process can be expensive. And so we need another alternative mode of producing this, which is hopefully more sustainable for the long term. And that brings us to my project, where I'm engineering yeast cells to produce Tanshinone IIA. Once you engineer a strain that can produce our compound of interest, which it does not naturally produce, we can really optimize and scale up production to increase yield and develop that more sustainable mode of getting this compound. And so you may be more familiar with the yeast I'm working with, Saccharomyces cerevisiae, under its more common name, baker's or brewer's yeast. So yes, this is the yeast that's capable of taking sugar and converting that into carbon dioxide and alcohol. And so we'll see this a lot of times being used in beer production or to raise our bread dough before we bake it. And sometimes what we don't often think about when we're doing these processes is that what we're tapping into here and what we're really taking advantage of is the yeast's central metabolism. And so this means that all of this production is very intricately linked to the yeast cell's growth. And as the yeast cell replicates, if we rewire those natural metabolic networks within the yeast cell and tap into even their specialized metabolism to obtain other targeted compounds that we've engineered them to make, we can link that growth with production of our compound of interest. And that's what we're trying to do in my project. And so just to recap what we've seen so far, number one, I love plants. And plants produce a bunch of specialized metabolites, all these compounds which we use already in our daily activities. 2, T2A is my target compound of interest, a compound that hasn't been tapped into yet in terms of large-scale production because of issues with obtaining it from its natural plant source. And 3, now we're going to be looking at how I'm engineering the yeast cells to produce T2A. So the first thing we need to kind of establish when we're discussing the production of any compound in a cell is how do we get from compound A to compound D? Well, if we think of it as a sort of factory's production line, you can imagine that within a cell there's all of these biological machines. And as compound A goes into the first machine, it'll come out as compound B. B will go into machine 2, come out as compound C, which will then go into the third machine and come out as compound D. And for those of you who might be more familiar with biological terminology, of course, these biological machines that I'm talking about are enzymes, which are a type of protein found within the cells. They take very specific compounds and shoot out other very specific compounds. They're a very precise approach. And specifically, the enzymes that I work with in my project, many of them require these specific environments in order to function correctly. In my case, lipid membranes. And you can think of membranes as just layers of fat that create separate compartments within a cell with very specific environments. And if you don't have those environments, the proteins will not fold correctly and they will not function correctly. Think of the effects if you were to take some milk and throw it into vinegar and it all curdles up. The same thing will happen to these proteins. They need this lipid environment. And so within a cell, as within our body as a whole, there are these individualized compartments called organelles, and they're typically bound by these lipid layers, creating these specific areas for specific types of production to occur. The most common membrane-bound organelles, which you might have heard of before, are the nucleus and the mitochondria. And what's really interesting is that if we compare a yeast cell to a plant cell, you'll notice that a lot of those membrane-bound organelles are present in both of the cells. The environments, in many ways, are quite similar. And the key difference there, and the reason why we prefer using yeast cells for production and scaling up of these compounds, is that yeast cells, or yeast as an organism, is a unicellular organism versus plants, which are multicellular. So where it might take a plant years, if not decades, to reach maturity and produce all of the compounds we're interested in, it'll only take a yeast cell 2 hours to reach maturity for 1 generation. So they grow really rapidly. And being a unicellular organism, it's also much easier for us to take a look at the metabolism, which is confined within that one cell versus spread throughout a whole plant, and engineer that and repurpose that. And so if we take a bit of a closer look at the T2A pathway and all the biological machine parts that we know are involved and those that we don't know, you'll see that there are 6 currently identified biological machine parts and 1 kind of black box in the process, where we don't really know exactly what's happening. And so we start with some 5-carbon compounds, which are present naturally in both yeast and plant cells. They go into the first machine, where they combine into 1 long 20-carbon compound. These go into the second machine, where 2 cyclic parts of the structure will begin to form. That goes into the third machine, which then adds on a 3rd ring to the compound. This will then go into the 4th one, where we get the addition of some decorative groups onto the structure. So these oftentimes in nature take the form of oxygens and hydrogens, and these are crucial for the function of that compound, for the biological activity. If these oxygens or these hydrogens are added on to the wrong spot or in the wrong orientation, it can completely change the activity of a compound, which is why it is so useful to have these biological machines that have that precision within them. That will then go through a next step where another compound, another carbon and some oxygens will be added on to the compound. Wait till we get there. There we go. And then we're not quite sure what's happening, but this group that's highlighted here, somehow within the cells, will then lose that, within the plant cells, will lose that group that was just added to produce our final precursor before T2A. And so we'll get into how we're working around that kind of black box of a step in just a moment. But first, let's take a bit of a closer look at some of the enzymes involved in the pathway, mainly just to emphasize how difficult it is to understand exactly what is occurring in that uncharacterized or that unknown step. So if we take a look at just one of the types of enzymes that I'm working with in these pathways, which is a type of cytochrome P450, you can see in the tree over here, you can imagine this is kind of a tree of species of different organisms, but it is all just cytochrome P450s. They are the largest class of enzymes present in plants. And the diversity of just that one class of enzymes is really reflected in its name. So in the same way that we would name animal species, we name genes and enzymes that they encode for. So you can see in CYP76AH24 that CYP corresponds to its superfamily, 76 the gene family, AH the subfamily, and 24 the individual gene. So there's a lot of cytochrome P450s, which can make understanding exactly what's happening in that portion a little bit difficult. And so in the first portion of my project, I transferred and collected all the genetic information encoding for the known biological machinery from the original plant organism into some yeast cells. This is typically done by putting the genes encoding for those enzymes into circular plasmids. These are just circular strands of DNA which facilitate that transfer of information. And we reconstructed the pathway of the early steps for T2A synthesis in the yeast cells. Once transformed with that genetic information, grew the yeast for a few days under some monitored conditions, and then we were able to measure using gas chromatography and mass spectrometry some of the compounds the yeast cells were producing. And each peak on this chromatogram over here corresponds to a different compound. And so from this, we could confirm that now the yeast cells were producing some early precursors in that pathway, which they did not naturally produce prior to our introduction of the biological machinery. And so, of course, so remember, there is that unknown step that we need to find a way to work around. And so our general approach for this portion of the project is to do something called in vivo directed evolution, which is essentially when you engineer the cells to perform evolution, which usually is over the course of thousands of years, but on a much smaller time scale because of their rapid growth. And by targeting this evolution with specific evolutionary pressures and growth systems that we've engineered for the cells, we can introduce enzymes as part of that pathway, biological machine parts that have been repurposed with little additions and little changes on their parts, different variants. And then the next steps of my project will be able to analyze some of those variants that we obtain and the different, using gene sequencing, more chromatographic analysis, and from there be able to determine if we've moved anywhere closer to T2A production in the yeast host. And so I just want to take a moment to really emphasize how incredible of a process this is, really, when we think about it. We've taken an organism, given it the information from another organism on how to produce something. Furthermore, this approach that we're developing is trying to find a way to work around the unknown parts of that pathway as well. And what's so incredible about fermentation with yeast and tapping into their metabolism is that you don't depend on these petroleum-derived chemical synthetic products. And you get that specificity from biological systems like plants and their machinery without any of the large land use and without any of that resource-intensive nature. And especially moving forward in the face of climate change, we need to think about the stability of our supplies of medications. When we're trying to produce therapeutics and preventative medications, we need to ensure that our mode of obtaining those compounds is stable. Otherwise, if they depend on environmental conditions and yields change year to year in our plants, prices will fluctuate as well. And it can create a lot of economic distress for those relying on those medications. And so I believe this form of fermentation, precision fermentation, is really key in building this resiliency we need in our communities moving forward in the face of climate change. Thank you.